In this video, we will talk about the reactions of the Calvin cycle, which is the second part of photosynthesis. The Calvin cycle is also called the dark reactions or the light-independent reactions. But these terms are really outdated and they can be misleading because the Calvin cycle reactions do not directly require light, but that doesn't mean that they only occur in the dark. They can actually occur in the dark or they can occur when the light is shining as well. They're also not totally independent of light because even though light is not directly required for the reactions of the Calvin cycle, we do need the products from the light reactions in order for the Calvin cycle to occur. So remember, in the light reactions, we produced ATP and NADPH, and those are needed in order for the Calvin cycle to occur. So indirectly, the Calvin cycle is still reliant on light energy because the light reactions have to occur first. The reactions of the Calvin cycle occur in the stroma of the chloroplast. So remember that a chloroplast is a double membrane bound organelle. And then inside we have these stacks called thylakoids. And remember, the thylakoids are where the light reactions take place. But inside the outer membranes, but outside the thylakoids, we have this fluid environment called the stroma. And the stroma is where the Calvin cycle reactions are occurring. So let's start by summarizing the Calvin cycle. So what happens in the Calvin cycle is that chemical energy in the form of ATP and NADPH, which were produced during the light reactions, gets converted to another kind of chemical energy, this time stored in the chemical bonds of carbohydrates. And carbohydrates are organic compounds that require carbon in order to be produced. And that carbon comes from carbon dioxide. We can break the Calvin cycle down into three main steps. The first step is known as carbon fixation. The second step is known as carbon dioxide reduction. And the third step is the regeneration of a molecule abbreviated as RUBP. On the next several pages, we will talk more about the reactions that happen during these main steps of the Calvin cycle to learn how these carbohydrates are made from molecules of carbon dioxide. At the beginning of the Calvin cycle, we have the reactions of carbon fixation. And what happens during carbon fixation is that a 5-carbon molecule known as RUBP combines with carbon dioxide, which has one carbon atom in it. In doing so, it forms a 6-carbon molecule. That ultimately gets broken into 2 3-carbon molecules called 3-PGA. So you'll notice that we are following the carbons here. So we took a 5-carbon compound and combined it with a 1-carbon compound to form a 6-carbon compound. We then took that 6-carbon compound and divided it into 2 3-carbon compounds. Now, RUBP stands for ribulose 1,5-biphosphate. But as long as you know RUBP, that is all you need to know for the sake of this course. As a reminder, CO2 stands for carbon dioxide. And then 3-PGA stands for 3-phosphoglycerate. Once again, for the sake of this course, as long as you know 3-PGA, you know all that you need to know regarding this compound. Now we can also draw this out by just using kind of black circles to indicate the carbon atoms. So remember that RUBP is a 5-carbon compound, and it combines with carbon dioxide, which has 1 carbon atom. In doing so, it produces a 6-carbon compound that gets broken down into 2 3-carbon compounds. So far what's happening is that we are just kind of combining and shuffling these carbon atoms around. Now, there are other atoms involved. So there are oxygens and hydrogens involved in this process, but the movement of the carbons is what we're really focusing on here. I should also note that these reactions are catalyzed by an enzyme known as RUBP carboxylase, which is also called Rubisco. The next step of the Calvin cycle is carbon dioxide reduction. Now, we're really talking about carbon reduction here because the carbon dioxide was fixed during the first step of carbon fixation. Now, remember that reduction is a type of reaction in which a molecule or an atom gains an electron. So if you remember our mnemonic of oil, rig, oil stands for oxidation is loss of electrons. Rake stands for reduction is gain of electrons. So during these reactions, the molecules 3-PGA that were produced during carbon fixation get converted to a molecule known as G3P. And G3P stands for glyceraldehyde 3-phosphate. And if you don't remember any of the names of the other abbreviated molecules, this is one that you should probably know because it's going to show up again in future processes that we talk about. Now, during this process, the 3-PGA molecule is being reduced, and so it's going to gain electrons, which leads us to the question of where do those electrons come from? And the answer is that it comes from NADPH. So remember that NADPH is this electron carrier that was made during the light reactions of photosynthesis. And NADPH is an electron carrier. And remember, I draw these little dots to kind of stand for the electrons that it is carrying. And so what happens is that NADPH gives those electrons to 3-PGA, and it gets converted back to NADP+. And so in that process, NADPH is being oxidized, meaning that it is losing electrons, whereas the 3-PGA molecule is picking up those electrons, so therefore it is being reduced. Now this reaction also requires energy in order to occur, which leads us to our next question of where does that energy come from? Well, the energy comes from the ATP that was also made during the light reactions of photosynthesis. And so ATP is broken down into ADP and phosphate. And in doing so, the energy is transferred to these molecules in order to facilitate this reaction. And once again, remember that the ATP came from the light reactions. And the NADPH also came from the light reactions. And then just another reminder that the 3-PGA came from the first part of the Calvin cycle. This came from the carbon fixation steps. Now, one molecule of G3P isn't very helpful to the plant. So the Calvin cycle actually has to happen 3 times in order to give us an amount of G3P that is really usable to the plant. So if we do 3 turns of the Calvin cycle, we end up producing 6 molecules of G3P. And these 6 molecules of G3P are important for the next steps of the Calvin cycle and for the ultimate production of carbohydrates. The third and final stage of the Calvin cycle is the regeneration of that molecule, RUBP. And this is important because remember that the Calvin cycle is a cycle. And the cycle actually starts and ends with RUBP. So it's important after that RUBP combined with the initial carbon dioxide to go through carbon fixation, at the end of the cycle, the RUBP has to be made again so that the cycle can keep happening. So how do we regenerate that RUBP is the next question. And the answer comes from those 6 G3P molecules that we made through carbon fixation and carbon reduction. So of those 6 G3P molecules, only one of them actually goes towards making the organic compounds or the sugars that we talked about originally. The remaining 5 G3P molecules actually go back into creating RUBP. The creation of RUBP requires an input of energy, and that energy once again comes from ATP. And remember that the ATP comes from the light reactions of photosynthesis. So let's do a quick review of the Calvin cycle. So remember that the Calvin cycle starts with this molecule known as RUBP. RUBP combines with carbon dioxide to form molecules known as 3-PGA. And this reaction is catalyzed by an enzyme known as RUBP carboxylase, which is also called Rubisco. The molecules of 3-PGA then get converted to molecules of G3P, and this requires an input of energy from ATP and an input of electrons from NADPH. With 3 turns of the cycle, we end up making 6 molecules of G3P. Of those 6 molecules, 5 molecules of G3P go into remaking the original molecule of RUBP so that the cycle can happen again and again. The remaining 1 molecule of G3P is then able to be converted into organic compounds, which includes sugars. Also recall that the regeneration of RUBP requires an input of energy in the form of ATP. And remember that the ATP molecules and the NADPH molecule come from the light reactions. And so we can divide this cycle into 3 sets of reactions. The first series of reactions is called carbon fixation. The second set of reactions is called carbon dioxide, or just carbon reduction. And remember that the 3-PGA molecules are being reduced while NADPH is being oxidized. The third set of reactions we can call the regeneration of RUBP. Let's talk a little bit more about this molecule that we called glyceraldehyde 3-phosphate. And remember, we abbreviate this as G3P. And this is a very versatile molecule. It's a carbon-based compound that can actually be converted into a variety of other compounds. For example, it can be converted into glucose, which is a monosaccharide. The glucose can then be combined with a fructose molecule, which is also a monosaccharide, to form the disaccharide known as sucrose. And sucrose is also just called table sugar. because this is the sugar that you keep in your house that you can bake with or add to your coffee. It's just the sugar that you have on your table. And, and as I mentioned, sucrose is a disaccharide. The glucose molecules could also be converted to different polysaccharides. So the glucose molecules can be combined to form starch or to form cellulose. And both of these are polysaccharides. Starch is a molecule that stores glucose molecules that can be used for energy production, and cellulose is a structural carbohydrate that allows cells to make things like their cell walls. In addition to making sugars, G3P can also be converted into a molecule known as a fatty acid. And then fatty acids can combine with a molecule known as glycerol in order to form lipids. And lipids include things like fats and oils. G3P can also be converted to form amino acids. And amino acids are the building blocks of proteins. So we can see that this G3P molecule, which is produced at the end of the carbon reduction stage of the Calvin cycle, is a very versatile carbon-based molecule that can be converted through a series of additional chemical reactions to form different organic compounds.